So today we are here with Dr. Marta Pratelli. She is the first author on a paper entitled Drug-induced change in transmitter identity is a shared mechanism generating cognitive deficits that was recently published in Nature Communications. So thank you for talking with me today.
Thank you, Amara. Thank you very much.
Yeah, so I just wanted to start off. So you work in the lab of Dr. Nicholas Spitzer.
Yes.
What is the main focus of the lab and how did you get into that lab?
So the main focus on the lab is a form of plasticity that is now widely documented, but not very often studied or considered. So just to give you an idea, when we talk about brain plasticity, we talk about the ability of our brain, the cells that constitute that, and the way they functions to adapt a response to the external environment. So how our experience, whatever we live, is changing the way our brain works. And Nick's lab is studying how sometimes changes in environmental experiences or environmental changes can affect the neurotransmitter expressed by certain neurons. What does it mean? This means that basically it's like if a neuron that generally neurons are communicating one with another, I like to make this example. You can see them as the lights in the traffic lights that control the traffic of information in our brain. So what happens if one of these neurons change? The type of transmitter it expresses is like if a traffic light is subdialing when you want to switch a red light, you switch a green one, to give you an example. And so I've always been interested in investigating how experience can shape our way of perceiving the world and on behaving. And I found Nick work particularly fascinating. And so I decided to join for my postdoc.
Okay, so what was your undergrad and graduate experience like? How did that bring you to neuroscience in general?
Yeah, so actually, as an undergrad, my undergraduate was in molecular biology, cellular and molecular biology, and as well as my master, because I studied back in Italy, when you generally have a bachelor and a master to
Mm.
have a full education. And then I did my but for my master, I volunteered in a lab that was doing neurobiology, investigating the serotonergic system and generating basic tools, like genetic tools, to be able to investigate how the brain work. And that was really fascinating. So I then remained to do my PhD in the same lab and investigating plasticity in serotonergic neurons, mostly changes in their morphology. But then I wanted to do more, to go more close to something that have a readout on behavior, and this what
Okay.
me to Nick and neurotransmitters.
Yeah. So it seems like you've been in neurotransmitters for a lot of your career and everything. So you're quite the expert.
Yeah.
So you were discussing neurotransmitter switching before.
Yes.
That's okay. Okay, because that's something that's a major theme of this paper, but I wasn't sure of that exactly. So why did you guys in this specific paper choose to focus neurotransmitter switching with the idea of in the context of addictive drugs?
So first of all, because this transmitter switching, so this ability of neurons to change the transmitter, they express sort of this light of the traffic light that change color, right? To make a simple example, this is something that generally occurs when you have a stimulus that changes neuronal activity in a region for a sustained period of time. Now, this is something that drugs, multiple drugs, despite even drugs that have different mechanisms of actions in the brain, cause. They change, can cause persistent changes in brain activity in specific brain regions. Also, there have been some evidences of changes in transmitter, despite there were not clear evidence at a single neuron level. This is a little technical. So there were basically preliminary data suggesting that this may be the case, but they were not studying putting this in the perspective of, okay, there is a neuron that is changing what it's doing.
Mm. That makes sense. So can you just generally describe the major findings of this paper and what makes it exciting?
Okay, so yes, be happy to. So basically, this paper, we saw that there were some neurons in the frontal cortex, like it's called the prelimbic cortex, is the equivalent of the frontal cortex in the human brain, that are normally excitatory. So are neurons that basically send the message go. And then at a certain point, in response to repeated exposure to drugs, they start to become inhibitory. So they start to release a transmitter that send the opposite message. So don't go. They become GABAergic. Now, um, it was interesting the fact that we saw this not in response to one specific drug, but to two drugs that are completely different in the way they acutely affect the brain. One is PCP, also known as angel dust, and one is methamphetamine, which is a stimulant. Also, we basically mimic some of the effect of drugs of abuse by stimulating dopamine from the ventral tegmental area using a protocol that had been shown to mimic in mouse models some of the effect of drug intake and development of addiction by other groups. And even this was sufficient to cause these neurons in the prefrontal cortex to change their transmitter. Now, you will say, then why is this important? What was very interesting about our finding is that we noticed that this change is needed for the appearance of memory deficits and cognitive impairments that occur after repeated exposure to the drug. Because if we give the drug, but basically we force the system, we manipulate it so that the neuron cannot change the transmitter they express, we no longer see the memory deficit and the cognitive deficit. And what if I can allow for example, what me and Nick were very enthusiastic about is the fact that not only we can go with we can manipulate gene expression to have this effect on behavior, but that the same effect was achieved by manipulating neuronal activity in the region. So now we do it with a classic neurobiological tool that is called chemogenetics. But we believe that this is important because there are so many studies now and also clinical trials like trying to use way of manipulating activity to cure different type of diseases, including addiction. Uh,
Mm-hmm.
and it may be interesting to know more about if and how these non-invasive stimulation methods can actually interfere with transmitter expression.
Yeah, I think that that is fair. So you talked about doing you had behavioral effects that you were able to like reverse with your manipulations. Can you
Yes.
talk a little bit more about like what the behavior you conducted was and like what type of memory you were testing that you saw deficits in?
Yes, of course. So we did a very simple, like the behavioral tests we did were fairly simple and well established. So it was nothing particularly complex. So one test is called novel object recognition. It measured the ability of retain memory. So the way it works is that you take a mouse and you expose it, like you put it in a place where it can explore, and you have two object, identical object that is gonna explore. Like mice are very curious, so if you expose them to two objects, they're gonna go there and interact with them and so on. Then the day after, you take the mouse again, you bring it in the same arena, you put one of the objects you have already seen plus a new one. Now, normally what happens is that the mouse is very curious, so it doesn't really care about the object it had interact with the day before, but it's curious about seeing the new one. So it's gonna go and explore the new object. Unless there is some deficit in the ability to or recalling what his experience had been the day before, in which case it's gonna explore both objects equally. So this is exactly what we see in response to drug, that we have this loss of ability to discriminate a stimulus that you have seen before from a stimulus you have not seen. And this is one of the tests. The other test is a working memory task. And it's a simple task that involves having a mouse moving into a multi-arms maze. And generally, according to the same principle, which says that principle, more than principle, according to the same um, ability, curiosity that is intrinsic in rodent models. When you put them in a maze with multiple arms, the mouse start to explore, go into an arm, then it change, go into another, then you move and explore another one. It generally, what it generally doesn't do very much is exploring an arm, go out and go back where it just went, because there is a seek of novelty. In mice that have been exposed to drugs, and this is not exposure one time, it's multiple exposure and then withdraw. So it's a long-term effect of chronic treatment. And what happened is that the mouse really gets a little bit confused of what he explored and he didn't explore. So he go in a maze, then he exit, then he go back. And so you have an impairment in his ability to alternate between the different arms of the maze to
Mhm.
equally explore the maze, but he tend to get confused when he arrive to change arm, basically.
Mhm. So is this type of, so one test was a working memory test and the other was a more long-term memory, like the 24 hour?
Long-term memory,
Yeah.
exactly. Yes.
So is this type of memory loss something that's recapitulated in the patient population?
It is sometimes lost, but there is not a quick, clear, let's say, there is not a clear the readout.
Okay.
Like, I'm not personally aware of any clear test recapitulating the same things.
Yeah.
But obvious that some of the effect that caused by long-term use specifically of this drug as compared to other drugs is to have a decline in cognitive function, including memory. But I cannot tell you if there is a specific, like I do not know at the top of my mind of a paper that have characterized a similar type of memory in humans.
Okay. Okay. So the one of your findings in the paper was that the memory loss wasn't from neuronal loss, like you weren't seeing apoptosis with this
Yes.
drug use, which I think is a common misnomer in real life. People think like, oh, you do drugs, you lose your neurons, but you actually found a much more complex mechanism that was something that was reversible, right?
Yes, exactly. And this is why exactly why it's more interesting, because it's not something that is a permanent loss. It's something
Mm-hmm.
that actually it seems more of an adaptation of function, for
Mm-hmm.
which since you have a stimulus that is a strong stimulus, like a change of the normal status of a change in the way the brain normally work, it causes these neurons to change the type of signal they see, but this can be reversed. And reversing it can have some beneficial effect on the underlying function.
So has this neurotransmitter switching like been seen in other models or in humans, or is this something unique to like the models that you use?
So no, this is not unique. Actually, we didn't look, I didn't have the chance to look in human samples, but there are other stimuli that cause the same type of switch. For example, acute stress, acute inescapable stress does it, and in that case, it's been reported both in has been also the same type of change in transmitter has been reported both in mouse models as well as in post-mortem human brains from patients with PTSD as compared to the population control. There are other evidence, like there are actually many evidence of different type of transmitter plasticity. Some of them are also are instead advantageous, like they can be beneficial. Like
Oh, okay.
for example, yeah, there has been evidence of running voluntarily physical activity causing a transitory, however. This is another characteristic. It seems that the maladaptive changes in transmitter are more stable over time, while the beneficial one seems not to be stable. They spontaneously reverse after a while, but they can occur in response to running to facilitate motor learning. This has been done in mice. There are also evidence that changes in the daily photo period, meaning the amount of light and dark you experience during your daily routine, cause changes in some transmitter in your hypothalamus. And this has been observed in rats, in mice, and in humans
Okay,
together.
so it's a very widespread, like conserved mechanism.
It's a conserved mechanism. It's still like that. I wish there were more people studying it so we can
Mm-hmm.
have more information, and it's still pretty scattered. Um, but yes, it's conserved.
So in your study, you did some pretty advanced techniques for reversing this, including like AAVs and that, but you also used clozapine.
Yes.
to reverse it, which is a drug that's used for as an antipsychotic for
Yes.
schizophrenia. But is this something that could be possible to be, used in the drugs of abuse field and, have a clinical trial for that type of treatment?
So, first of all, I wanna like this is actually an important details that bring us back to the memory deficits. Like why these the one of the like the in mice PCP or methamphetamine, especially PCP. So the drugs are used are able to use to model some aspect of schizophrenia-like behavior that has been associated exactly to the type of memory deficit I'm also observing. And this was why we wanted to test the effect of an antipsychotic that was shown to be effective in curing some of the cognitive deficit observed in some cases of schizophrenia. And it turned out to work through the same mechanism. And in terms of whether clozapine itself can enter of a clinical trial for addiction, I think it can be tricky because clozapine itself have some severe side effect.
Mm.
And the way that we are thinking about it now is but it's good as a proof of principle to see that some pharmaceutical can have this effect. But since clozapine is a very dirty drug, what we are trying to do now is to study instead what are the neuromodulators and transmitter that clozapine is acting on to see whether the same mechanism can be achieved with a less um, dirty drug and maybe with a treatment that have less side effects.
Okay. That's very, so is your lab specifically working on that, or are you like working with other labs to do something like that?
So this is very at the very beginning, meaning
Okay.
that I'm establishing now a series of fiber like for the technical fiber photometry to measure the release of neuromodulators, just to
Okay.
have some preliminary data and then seek
Yeah.
further collaborations on the
No,
on.
that's very exciting though. That's still, a good next step.
Yeah,
I'm just gonna finish off with a more broad question. So,
of
what are
course.
some exciting research things happening in your field, and what areas still need to be developed that you're excited
Oh, many.
about?
It's a very broad. So,
Yeah.
first of all, I want to talk about this because I recently dig into that to write a review, and so it's particularly fresh. Like, actually, the ability of drug to change transmitter in the brain seems to be more vast than what we generally think about, meaning that just looking around, even if nobody maybe used or the majority of the study doesn't definitely talk about transmitter switching or transmitter plasticity, there are evidences that many systems and many regions can change in response to drug stimuli.
Mm.
Now, since those are reversible changes, I will be very excited to see more research into this topic, or even just like that when um, a researcher stumble into something that look like a change in neurons expressing a transmitter, they just don't disregard it.
Hmm.
A
Yeah.
study would work because it can be an important part of a mechanism. And so I'll be excited into that. I'm particularly very excited to see the progress that are going on very quickly in terms of different ways to stimulate and modulate neuronal activity with non-invasive approaches, because it's definitely a new way to look at therapies and generally are safe.
Yeah. And translatable. Yeah. No, that's definitely
Translatable,
fair.
safe, and I think it's an interesting new set of data that it's a set of expertise.
Hmm.
So I'm looking very favorable at changes that are going in that direction. And then in general, in the field of drug abuse, I think, despite my work didn't get to that level yet, but it's definitely where I want to bring it. Um,
Hmm.
I think it's important to consider the studies that are including multiple aspects, not only the drugs, but the drugs and the social interactions, for example, because it is be like, or the quality of the environment in which the mouse lives. So I think everything that try to connect not only the effect of a drug, but an effect of an environment and experience. I think it's are interesting new aspects that are coming to the table.
Yeah, I think that is fair. I also think like having studies like yours where like it discusses transmitter switching will really, I feel like people report things they've seen but they don't have a name for it, so they just don't trust it, or and like when they can get the name they're like, "Oh, that's what I was seeing."
No, exactly. Thank you for bringing it up because that's exactly why I'm very happy also to discuss this. I don't, um, it's just that, it's something that you see people notice. You read a paper and you find it sometimes, but it generally put on a side because there is not a name, is not considered. So it's good to have it and to pay attention because
Yeah.
some of them may not be consequential.
Yeah. And it's a
Um,
new opportunity for things like translatable drugs. Like that,
exactly.
if that's something that modulates it, that's a great opportunity to look into in the future. So,
Exactly. So considering also that maybe we identify 10 and of that 10, one is important. Like,
mhm.
but if we keep our eyes open, this can be an interesting phenomenon on which we can intervene through different approaches.
Yeah. Well, thank you so much for your time.
No, thank you so much, Amara. I
Yeah.
appreciate.
Yeah.